Good afternoon, everybody, and welcome to our second lesson on genetics. Today we're going to be talking a little bit about DNA and how DNA gets copied and moved out of the nucleus to actually be used. But we're going to start off just by breaking down DNA as a substance, right? DNA is in our nucleus. It contains all of the information that you need to be a living person, or all of the DNA that corn needs to be a piece of corn. And all of that information is stored based on nitrogen bases, right? The four nucleotides that make up DNA are adenine, thymine, cytosine, and guanine. Right? And these four bases pair together, and the order that they come in, right, G, T, T, C, this order that they come in is the code that then becomes proteins. In the double helix, every nucleotide is paired, right? Of the four, we have two pairs. A's and T's pair together, and C's and G's pair together. I don't know why I'm circling all those. Right, A's and T's pair together, and C's and G's pair together. You'll never see a C with an A. You will never see a C with a T. You will never see a G with an A or a G with a T. Right? The only pairs are A and T, T and A, or C and G, G and C. Right? These nitrogenous bases, the G, T, A, and C, are our nitrogenous bases, while the phosphate and the pentose kind of hold together the rest of the DNA. That's this ribbon here is made up of these parts of the DNA, which you will learn more about in AP Bio if you ever decide to go into AP Bio. The nitrogenous bases are held together, right? It's not a permanent holding together of these things, right? It's almost like a zipper where they're held together. It's strong while it's held together, but it can be taken apart. You can separate the pairs from each other to access the information stored inside, right? And this thing that holds these bases together is called a hydrogen bond, which is something you're going to learn a lot more about in chemistry. It's a very, very strong, think of it like a very, very strong magnet holds these bases together. It's not going to just fall apart on its own, but if you use the right thing or use enough force, you can pull it apart and put it back together, and pull it apart and put it back together, and pull it apart and put it back together. Right? When you want to use your DNA to make proteins, you're going to open up your DNA, unwind it a little bit to access the bases that are in it, and then close it back up for storage. Right? Kind of like opening a recipe book to read the recipe and then closing it when you're done. When DNA replicates, it unwinds, it breaks apart, and then you build a new double helix around it, right? You take it apart and you add bases. Where did I put my pen? You take it apart and you add onto it the other bases to complete it, so that when you're done, you're going to have two copies, right? You're going to already see it starting, right? You're going to have two copies of the same strand of DNA. And this is how, during mitosis, you're duplicating your DNA, right? It unwinds and it builds the other half around it. So you end up from one double helix to two double helixes. But we're not really focusing on replicating our DNA. We care really about using DNA to make proteins. The DNA, the order is not random. The order of the DNA in groups of three, very specifically groups of three, code for amino acids. So when you're going to build proteins, right, say I want to build a protein, every amino acid that I want to put together is 3 nucleotides. Right? So TAG codes for an amino acid. CCG codes for an amino acid. ACT codes for an amino acid. And so in our DNA, you see these triplicates, right? These groups that repeat to choose the different amino acids to build the proteins that we then use. There are also 3 nucleotide codes for starting and stopping. So you know this is where the protein starts. These are all of the amino acids I'm going to need, and then this is where it stops. So of the amino acids that then come next, you can differentiate between this is one protein because it's between the start and the stop, and this is another protein between a start and a stop. Now we've got tons and tons of DNA in our body, right? Every cell has all of the DNA that every other cell in the body has. We need to get that DNA out of the nucleus to the places like the ribosomes where we can actually build proteins. And so, keeping with our book analogy, right? Imagine you need 1 page from the world's biggest recipe book. You're not going to take the recipe book home from you, right? It's a bajillion pages, right? It's got every recipe ever in it. So instead of taking the whole book with you home, cooking, and then bringing the whole book and putting it back in the library, you are going to make a photocopy of the 1 page that you need or the 2 pages that you need, right? I want to cook lasagna. So I'm going to photocopy the page for lasagna. I'm going to bring it home and I'm going to use that recipe to make my lasagna. Right? I haven't damaged the book. I just took what I needed from the book for that day. If I want to make onion soup another day, I'll go and I'll photocopy the page for onion soup and I'll bring that home instead. Right? mRNA, messenger RNA, is our photocopy machine. Right? DNA can't leave the nucleus. So when we want to build a protein, or when the body says, hey, you need to build a protein, you're going to get that by copying the DNA with the mRNA, and then the mRNA can leave the nucleus, go to the ribosomes, and give the code to make your protein. RNA is a little different than DNA. Mostly because the T in DNA is replaced by a U. Right, so it's the same 4 bases, except instead of this T, you have a U, and that's so that the body has a way of recognizing the difference between RNA versus DNA. Right, so RNA is AUCG, and DNA is ATCG. It's a direct swap. Nothing else changes, right? The A and the T still pair together. The C and the G still pair together. Here it's an A and a U, and the C and the G that pair together. It's just a swap for U and T. Again, so that our body and our cells can tell the difference. Say like, oh, you've got a U, that's RNA. Oh, that's got a T, that's DNA. Right? To identify those differences. So like I said, the mRNA is going to copy the information and bring it to the ribosomes. And that process is called transcription. Very similar to what I showed you for DNA replication, the DNA is going to unwind and open up. The RNA is going to be built around. Let's make it green, right? The RNA is going to be built around the strand of DNA, and then it's going to leave. It doesn't need to stay in the nucleus. It can do whatever it likes, right? So in the nucleus, You make that copy, and then the mRNA leaves. And that whole process is the process of transcription. Right? Think of the word transcribing. Right? If you're copying something, that's a transcription. So we're copying the DNA and bringing it out to the ribosomes. Then, outside of the DNA, at the ribosomes, this piece of mRNA gets read and used. Right? So the ribosomes read the mRNA, and they say, well, this group of 3 is this amino acid, this group of 3 is this other amino acid, this group of 3 is this amino acid, and they translate the mRNA into a protein. Right? So it's make copy, build the thing. The same way if we're looking at our recipe analogy, it's photocopy the recipe you want, cook the recipe. We're going to look a little bit more into the process of both of these things, but we're going to really focus on transfer RNA for transcription. Right? Or no, sorry, for translation. Jesus. Oh, it is Star Wars Day today, isn't it? With all the lockdown stuff, I barely keep track of days, right? May the 4th be with you all. So when we're looking at actually building the protein, we have a string of mRNA, right? The mRNA, which we're going to keep green, right? Our mRNA is a copy, right? A, U, C, C, G, G, U, U, A, C, U. Right, so this is a copy of our DNA that goes over to the ribosomes. So at the ribosome, this piece of mRNA needs to be read and turned into a protein. Right? And that's where tRNA comes in. Transfer RNA is a group of nucleotides that is directly attached to an amino acid. And so they come in groups of 3. I have a really good picture on the next slide that I want to show you. And the idea here is that the tRNA will attach in groups of 3. So I'm going to change color here to blue. It'll attach here U, A, G, and there will be an amino acid on the end. G, C, C, and there will be an amino acid on the end. A, U, and there will be an amino acid on the end. And so on and so forth. And then we're going to take those amino acids and link them together. Right? So again, this is what we're looking at. We're looking at our mRNA. This is copied from the nucleus. And then these pieces here, these are all our tRNA. They're going to bring these amino acids and line them up to make the correct protein from this recipe. And so you can see kind of how they're going to line up. We're looking here, we need a U, an A, and a C. And so this tRNA is going to click right here onto this trio match up and say, hey, that's where this amino acid goes. And the same thing for the rest. So when they move, they're going to line up, right? A's with U's, U's with A's, G's with C's, and C's with G's. Right? They're going to pair up according to the way they're supposed to be paired up. And we're going to build our protein based on these amino acids in this order. Right? Because remember, like we talked about in the very, very beginning of the year, order determines structure, structure determines function. All right, so by putting these amino acids in these order, or these amino acids in this order, we get a very specific protein and a very specific set of amino acids that will then work to do, I don't know, whatever, be a receptor for insulin, be an enzyme that breaks down starches, right? Just an enzyme or some protein or some receptor that we need in our body. Nope, sorry, not what I wanted to do. That's not the next slide button. This is the next slide button. So, like I said before, there are very specific codes for DNA, right? So each group, each triplicate is going to lead you to an amino acid, right? There are duplicates, right? So anything here that starts with a GG is going to be, don't remember all my amino acids, but it's going to read, there's only 20 different amino acids, right? 20 amino acids. And so all of them are coded into the DNA, right? Including things for stop codes, right? So if you read up until you get a stop code, right, you're going to get amino acids and then a stop code and then more amino acids and then a stop code, so that you know that this is one amino acid and this is a separate amino acid, and one protein and a separate protein, as opposed to it all being one big protein. And when we talk about problems that come up with DNA, one of the big problems that we'll see is either this stop code being messed up and not being read, or the stop code being skipped over when you're making your mRNA, so you get a bigger protein that probably doesn't do the thing you want it to do. And that's leading us into the idea of mistakes and mutations, which is what we're going to talk about next time. Right? So we'll talk more about the 4 different types of mutations later this week in our next video for living environment. Bear in mind that we are doing only Monday, Wednesdays now, because we are ahead of other groups. So only Monday, Wednesdays we're going to have class. Tuesday, Thursday, Friday is going to be office hours only. And there's office hours after class every day. I'm going to stop us here for now and transition us into office hours. If you have any questions, comments, or concerns, please stop by. And I hope you enjoy Star Wars.